So we have here the 5 microorganisms, which are the Enterobacter, Staphylococcus, Clostridium, Bacillus, and Listeria. So the first microorganism is the Enterobacter. So its first source is the product. So product, Enterobacteriaceae have been associated with the spoilage of a range of other foods and drinks, although many of these have not been extensively studied, and their exact role in the spoilage of these products remains unclear. Examples include beer spoilage by Obisum-Bacterium pertussis, spoilage of maple syrup by various members of the Enterobacteriaceae, spoilage of fresh creams deserved by Serratia spp and other members of the Enterobacteriaceae, which is characterized by clotting cream or from acid production and gas production. So, in shell eggs, Enterobacteriaceae are responsible for bacterial rot, with Enterobacter and Proteus associated with causing black rot. So the second source is the tract. So in the tract, the normal habitat of Enterobacter spp is probably soil and water, but the organisms are occasionally found in human feces and the respiratory tract. Infection of hospital patients, notably of the urinary tract, occurs. Enterobacter naturally inhabit the human intestinal tract, so water, animals, plants, and are common in foods. They are the cause of various nosocomial infections in immunocompromised patients. So Enterobacter species have an intrinsic resistance to ampicillin and cephalosporins. This enables them to survive and overgrow in the intestines of individuals treated with these antibiotics. And the third source is the utensils. So in utensils, kitchen sponges are a major source of cross-contamination as they can transfer foodborne pathogens, infectious agents, and spoilage, causing microorganisms to food contact surfaces. The effect of storage, which is 3 and 10 days in the microbial population of kitchen sponges at room temperature, which is 21 degrees Celsius, was assessed. Enterobacteriaceae isolated from sponges were identified and their antibiotic resistance determined. Kitchen sponges are one of the main cleaning tools used to clean kitchen utensils and surfaces such as kitchen utensils, cutting boards, sinks, oven tops, and refrigerators. However, during cleaning, food residues may adhere to the sponge surface, and damp sites such as sink areas can act as further microbial reservoir that can contaminate the sponges during their use. So the second microorganism is the Staphylococcus. So in Staphylococcus, the first source is the handlers. So in handlers, the presence of the staphylococcal enterotoxin is usually due to cross-contamination of ready-to-eat food with either raw or most likely contamination from a food handler that is carrying staphylococcal aureus. So food can become contaminated during the preparation if the food handler is a carrier of Staphylococcus aureus, and this is transferred to the food through direct contact with contaminated skin or by coughing and sneezing. Source of the Staphylococcus is the product. So staphylococcal food poisoning is an illness of the bowels that causes nausea, so vomiting, diarrhea, and dehydration, as well as ma-dehydrate ka. It's caused by eating foods contaminated with toxins produced by Staphylococcus aureus rather than a true infection with the bacteria. The third source of Staphylococcus is the urinary tract. So in urinary tract, Staphylococcus aureus is a cause of urinary tract infection among patients with urinary tract catheterization. The majority of isolates are methicillin-resistant Staphylococcus aureus. So, Staphylococcus aureus bacteriuria can lead to subsequent invasive infection. The efficacy of antistaphylococcal therapy is preventing late-onset staphylococcal infection in patients with persistent staphylococcal bacteriuria should be tested in controlled trials. The third microorganism is the Clostridium. So there are three sources. The first source is the product. So in Clostridium, in products, in most cases, Clostridium perfringens food poisoning results when you eat improperly cooked and stored foods. So normally, bacteria are found on food after cooking, and this bacteria can multiply and can cause Clostridium perfringens food poisoning if the food set out and cool before refrigerating. So commonly infected foods include meats, meat products, and gravy. The second source of Clostridium is the intestinal tract. So in intestinal tract, Clostridium perfringens can be found in the intestines of animals. So it can be found in animals. So this bacteria makes spores which act like protective coatings that help the bacteria to survive. So they help the bacteria to survive under certain conditions such as when the animal intestines is kept at unsafe temperature. So after someone swallows the bacteria, it can produce a toxin or a poison that causes diarrhea. The third source of Clostridium is the feed. So in the feeds, broiler feed can include meat and bone animal meal and vegetable meal. And one of the major contaminants of these feed components can be Clostridium perfringens. So it is Clostridium perfringens because chemicals have the potential to inhibit the growth of this pathogen. So in poultry, Clostridium perfringens is the pathogen responsible for the disease known as necrotic enteritis that causes economic losses. So these losses can reach 33% of chicken production in a broiler farm due to decreased nutrient absorption, leading to lower weight gain and feed conversion. So the fourth microorganism is the bacillus. So the first source of bacillus is the dust. So in dust, bacillus species can form dormant spores under adverse environmental conditions. So these endospores may remain viable for long periods of time. Endospores are resistant to heat, chemicals, and sunlight and are widely distributed in nature, primarily in soil from which they invade dust particles. So some types of bacillus bacteria are harmful to humans, plants, or other organisms. So most strains of bacillus are not pathogenic for humans but may also as soil organisms infect humans accidentally. So it can really infect humans incidentally. And the next source of bacillus is the products. So, in products, if you ever thought you had 24-hour flu, you may have actually had a foodborne illness caused by the bacteria Bacillus cereus. Foods that have not been properly stored at safe cold or high temperatures are the main sources of the pathogen. Cool foods should be kept cold below 40 degrees Fahrenheit, even during food service, and hot food should be hot, which means above 140 degrees Fahrenheit. Proper temperature control can be a problem when large quantities of food are prepared in refrigeration space. So, the third source of bacillus is the water. So, in water, the behavior of the sporulating swirl-dwelling Bacillus cereus, sensualator Bacillus cereus, which includes foodborne pathogenic strains, has been extensively studied in relation to its various animal hosts. The aim of this environmental study was to investigate the water compartments from rain and soil water as well as groundwater, closely linked to the primary Bacillus cereus reservoir for which available data are limited. Bacillus cereus was present primarily as spores in all of the tested compartments of an agricultural site, including water from rain to groundwater through salt. The last microorganism is the Listeria. So the first source of Listeria is the products. Listeria is found in refrigerated, ready-to-eat foods such as hot dogs, deli mix, and pasteurized milk, raw sprouts, dairy products, and raw and undercooked meat. And the second source of the Listeria is the handlers. So, food handlers were positive for Listeria monocytogenes, and a statistically significant association was found between the presence of these microorganisms and the following risk factors. And the third source of Listeria is the utensils. So, in utensils, places on your utensils where Listeria monocytogenes might start building up without being noticed easily are typically those places that need manual cleaning, having crevices, and often have standing water or show the marks of standing water like calcite deposits.